All right. We know from previous studies that exposure to particulate matter, or PM, is associated with increased hospital admissions and premature death from cardiovascular disease, as well as accelerated progression of atherosclerosis. However, we don't know what characteristics of PM are responsible for these effects. For example, the National Research Council listed as one of the gaps in PM research the investigation of PM's toxicity with respect to particle size and its constituents. Also, more recently, the Health Effects Institute released a report that stated that more research was needed on controlled animal exposures to ultrafine PM, and that cardiovascular effects were of particular interest. Dr. Kleinman's study has addressed several of these research gaps. His research investigated toxicity of ultrafine PM and a subset of its components, as recommended by the NRC. Also, his study examined cardiovascular effects using controlled animal exposures, which addresses HEI's recommendation. Dr. Kleinman's project is valuable to ARB for a number of reasons. The research was conducted in Los Angeles, so it has particular relevance to California traffic exposures. In addition, Dr. Kleinman's findings add to our overall knowledge base on ultrafine PM health impacts. His results provide insights into the role of ultrafine PM in accelerating the progression of heart disease, as well as the role of semi-volatile constituents in PM toxicity. These findings will help inform ARB in its efforts to protect the health of all Californians. And now it is my great pleasure to introduce today's speaker, Dr. Michael Kleinman. And I'm going to read a little bio for you first. Okay. Dr. Kleinman has been studying the health effects from air pollution exposures for over 30 years. He's currently professor and co-director of the Air Pollution Health Effects Laboratory in the Department of Medicine at the University of California, Irvine. He earned his PhD in Environmental Health Sciences from New York University and an MS in chemistry from the Polytechnic Institute of Brooklyn. Dr. Kleinman has published more than 100 peer-reviewed journal articles on a diversity of topics, including the uptake and dosimetry of inhaled pollutants, and health impacts from acidic and non-acidic aerosol exposures, as well as mixtures of particles with other airborne pollutants. His previous studies have examined cardiopulmonary effects of concentrated particles in animal models of susceptible human populations. He's currently investigating the role of organic and inorganic constituents of air pollution in the exacerbation of heart disease, which you'll hear more about in today's seminar. Dr. Kleinman is a consultant to the U.S. EPA Science Advisory Board, a member of the Board of Scientific Counselors for the Centers of Disease Control and Prevention, and chair of the California Air Quality Advisory Committee. Additionally, we are very pleased to announce that he has just been named chairman of the State of California's Scientific Review Panel on Toxic Air Contaminants. So on behalf of ARB, I'd like to offer our congratulations on this appointment. So without further ado, please help me welcome today's speaker, Dr. Michael Kleinman.
Well, thank you very much. It's really a pleasure to be here and be able to present some of these research results. And hopefully, I'm going to move this. Hopefully, what we'll do is go through the rationale behind the project, some of the methods we're using, so you can get a full picture of the intricacies of doing this. But I think the research results are very clear and are very important. So what we'll do is we'll talk a little bit briefly about the origin of the volatile and non-volatile components that make up ambient particles and looking at it from the standpoint of how they're formed during combustion. So this is relevant not only to motor vehicles, but to other types of combustion processes, power generation, and boiler operations and things like that. So it's not just limited to mobile sources. However, in Southern California, the major source of ultrafine particles appears to be related to mobile sources. We'll talk a little bit about how exposure occurs and what that means in terms of where in the respiratory tract particles are going to deposit. I also want to point out some of the characteristics of what is being called black carbon and brown carbon. And because there is a fair amount of confusion about it, and different scientific disciplines look at these things somewhat differently. So I'm going to try to put it into just a framework that applies to what I'm doing, and so that we're all on the same page. Then I want to talk a little bit about the potential mechanisms, how these particles might affect health, and then get into the details of the cardiovascular health effects study that I did, and we'll talk about the methods, the results, and some of the conclusions. And at the end, I'm going to put up some slides to acknowledge all the other people that helped with this, as well as the research sponsors. So don't go away before you see that. One of the things that I think is important to consider is a lot of what we know about ambient nanoparticles really comes from what we've studied with traffic-related particulate matter. And the diagram we have here shows an idealized picture of what you might see at some time after particles are emitted. And in this particular case, we have 4 peaks. The peaks that are in the very fine particle region, or particles smaller than 100 nanometers or 0.1 micrometers, are termed ultrafine particles. But even within the ultrafine particle fraction, there are particles that are somewhat larger and somewhat smaller. These guys down here, the very small particles, are the ones that are forming by nucleation of gases to form particles condensing in the air. These particles, let me try it with this. There we go. These particles react very rapidly and on contact with each other, and they form a larger mode. And so the ultrafine particles can be very small, down at the 10 to 1 nanometer size. But these don't last very long. They're very short-lived, and they will grow and form larger particles. We also have a condensation mode, which probably has a mean diameter up around 400 nanometers or 500 nanometers. And these are formed by particles in these smaller regions colliding with other fine particles, and they grow into these larger mode particles. The really large particles, the 10 micron and larger, and even down as low as 1 or 2 microns, are particles that are generated differently. These are generated more from mechanical processes. And so it's very difficult to mechanically break up a large particle into something much smaller than 1 micrometer. And so when you look at the particles in the atmosphere, you'll find this larger mode, which contains most of what we're calling PM10, which are the particles from 10 microns on down. So the larger frame, or the coarse particle fraction, are generated more by mechanical processes. The finer modes are produced more by combustion processes or atmospheric chemistry. To give you an idea of size, relative size, this would be what a PM10 particle might be sized at. And to give it a more physical form, if you've ever looked at a microscope and seen a red blood cell, a 10 micron particle is about the same size as a red blood cell. The particles that are PM 2.5 are actually 2.5 diameter, are, as you can see, considerably smaller as you get down to what we're calling ultrafine particles below 100 nanometers, and then we get down to the nanometer size particles. And so relatively speaking, the 10 nanometer particle would be this little dot, and you really can't see the 1 nanometer particle. In fact, with a light microscope, you're very hard pressed to see particles smaller than, I would say, 2 microns. Maybe sometimes you can push to 1 micron. You certainly can't see, under normal preparations, an ultrafine particle. And so all of the work that's done trying to visualize these particles uses electron microscopy techniques. Black carbon and brown carbon aerosols, they're very important constituents of PM 2.5 and of ultrafine particles, which I'm going to abbreviate as UFP. And for the purpose of the talk, and especially for the biological studies we're doing, what we're calling ultrafine particles are really particles that are smaller than about 180 nanometers. That's one of the physical constraints of the system that we're using. So it's very hard to selectively have particles smaller than that in a controlled manner. So black carbon, when you look at a filter that's collected in almost any urban environment, and you collect that filter for several hours, it will be black. So the black carbon is mostly elemental or graphitic type carbon. It is a very strong light absorbing material, and very often when we talk about black carbon in the atmosphere, we're talking about the amount of material absorbing light. So it's really defined by light absorbing characteristics. It's formed by incomplete combustion of fossil fuels, biofuels, burning of biomass. There are many different sources. So black carbon is emitted directly into the atmosphere, and usually in the form of fine particles. But a large fraction of that are in the ultrafine particle fraction, and they would be considered nanoparticles. Black carbon is the most effective form of absorbing solar energy. So it has an impact on global climate change and other characteristics. However, black carbon, being a major component of soot, is not present by itself. Very often, the black carbon particles are coated with organic materials. And those organic materials can also, to some extent, absorb light. And so when we look at atmospheric particles, we'll see a mixture of solid core particles that have a coating of organic materials on them. And sometimes we'll also see organic liquid droplets. And these organic compounds also absorb light, and atmospheric scientists have come to call those brown carbon. For a lot of the purposes of the toxicological work that goes on, we collect the samples and we measure them as a mixture of black carbon and brown carbon, And we separate it into a category called elemental carbon, which is primarily just the core particle, and organic carbon, which is the oily material that's either attached to the particle or present as independent drops. Now, in order to understand the way that these particles can affect health, we really need to look at how do you get them into your body. And so that really begins with the respiratory tract. And the respiratory tract is not just the lungs. The upper respiratory tract, including the nose, the mouth, the pharynx, and the upper tracheal tubes, are part of the respiratory system, and particles that deposit in the upper respiratory tract can still have biological effects. The smaller particles can penetrate down through the trachea, through the bronchi, and through the smaller bronchi, eventually getting into bronchioles and then into the alveolar section of the lung. So the lung is a very complex structure. Compared to an organ like the liver is an organ that processes a huge amount, almost, I would say, 90% of the food that you ingest and the toxic chemicals associated with the food that you ingest. All of these things are processed in the liver. The liver has approximately 4 different types of cells. The lung has something close to 45 types of cell. So the lung is a very complicated structure, both physically and biologically. When we look at the sources of black carbon, if we look globally, the overwhelming source is power generation and biomass burning. But when we look at the US emissions, transport processes make up more than half. So for most of the black carbon that we're looking at, especially in Southern California, we're really seeing a large impact from the black carbon from motor vehicles and off-road vehicles. We also have some biomass burning, usually inadvertently from wildfires, but these are sporadic events. Over the last several years, the amount of mobile source emissions of black carbon materials has been diminishing, which and is projected to continue to diminish as we improve control strategies. So overall, we've made some very good progress at removing a lot of the contaminants from the air. So what I'm going to be showing you is, despite the fact that we've removed a lot of these contaminants from the air, there are still residual materials that are associated with causing effects on health. Where do these compounds come from? They really occur during the combustion process, in which you're taking a hydrocarbon material. It could be coal, it could be fuel oil, and you're combusting that in a flame zone. During the combustion process, materials are oxidized to some extent, but some material is reduced. And so you're taking hydrocarbons, which are carbon-hydrogen molecules, removing everything and condensing it back down to elemental carbon particles. And so you get particles that are formed by charring processes. And these char fragments can be emitted and come out as fly ash, but some of them come out as soot in the nanometer size range and can react with organic materials from the vapor phase. So the some of the combustion products are organic vapors. Those organic vapors form in the flame along with a lot of inorganic materials, and they can form very complex organic molecules. So you can go from very simple hydrocarbon materials and build up to polycyclic aromatic hydrocarbons. Plus, you can get brominated and chlorinated and nitrated hydrocarbons. My fingers are a little too sensitive on this. But at any rate, what happens is that these nanoparticles become coated with organic molecules. In addition, there are transition metals that are part of the contaminant products in the original fuel that are either volatilized by heat or sometimes just emitted during the combustion process. And these can also react and act as catalytic sites. And so the net result is you form large fractions of compounds that we know are, at least in chemical testing, are quite toxic. So polycyclic aromatic hydrocarbons and the oxy polycyclics, nitroarenes, as well as others. And all of these things can coat the particles. When you look at a plume coming out of a stack, it's not a stagnant process. You don't emit something and it blows downwind. So this is an interesting picture. It's done by LiDAR. And what they're looking at is a color map. So as we're going from the dark blues on up to the yellows and oranges and reds, we're increasing particle number. So we're going from a few hundred particles per cc up to hundreds of thousands of particles per cc. And what you can see that's really interesting, contrary to what you sometimes think of as plume dynamics, where the plume goes out and it just tends to diffuse and get lower in concentration as it goes further away. The particles actually grow in. And so materials that were warm, organic vapors, are condensing as the plume moves downstream. And you can see that the particle concentrations in the plume tend to get higher and higher. So one important take-home message is that the characteristics of the pollution that people are exposed to are not necessarily the characteristics of what comes immediately out of the tailpipe or the smokestack. These things are going to react in the atmosphere, change, and we will have different products kilometers away from the original source. When the particles get inhaled, and this is a very simplistic view of it, but the blue dots represent bigger particles, the coarse particles, bigger than 2.5 microns. And they tend to deposit more in the bronchial tree. The fine particles can penetrate deeper down and move down into the smaller airways and eventually into the alveoli. The alveoli are actually the only working part of the lung because the alveolus is the place where oxygen is brought in from the air, moves across the alveolar wall into the bloodstream, and CO2 moves out of the bloodstream into the alveolar airspace, and then you exhale it. So on every breath, air comes in, oxygen transports into the alveolus, through the alveolar walls into the blood. So any damage to the alveolar wall structure or to the process of bringing air in by stiffening the bronchioles or airways can reduce the amount of oxygen you take in and the amount of oxygen available to feed your brain, to feed your muscles, things like that. And the way that they deposit by particle size, this is something that can give you a bit of an idea of what goes on, but as a function of particles, the respirable particles, which are typically the ones smaller than about 5 microns. These are the ones that will penetrate through the upper airways and really have a major impact on the respiratory tract, the lower respiratory tract. You can see that when you get down below about 4 microns, and this is for humans, you're getting about half of the material that comes in deposits. And when you get to the really small particles, almost 100% deposits. What this graph doesn't show you is the nanoparticles, because nanoparticles deposit by a different mechanism than the bigger particles. Nanoparticles diffuse. What that means is these particles are so small that vibrating air molecules in their path have enough energy to actually make them move. And so rather than moving with the airstream, they bounce around, and so they have more of a potential for hitting wall surfaces along the airways. One of the important considerations for this is that a substantial number of nanoparticles actually deposit in the nose when you're breathing them. The rest of them do actually penetrate down into the deep lung. So nanoparticles are a little bit different than the larger particles in where they deposit. But when they get down into the lung, they don't necessarily stay there. So this is a picture of the lung of a person who lived in Los Angeles for roughly 80 years. And what you can see, the blue arrow is pointing to, are black deposits. And these are elemental carbon as well as other insoluble materials that actually came in through the respiratory tract, deposited in the alveolar airspace, were picked up by macrophages from the innate immune system. These macrophages actually patrol each alveolus and pick up deposited material. Their main job is to eat bacteria and viruses and kill them. But if they pick up something insoluble, they can't kill it. And so what they do is they take it out of the lung so that this material doesn't accumulate inside the very important alveolar airspace. So where does it go? Well, some of these macrophages can literally crawl back up through the respiratory tract and come back up into your throat, and you cough it out or swallow it. But a lot of it, the macrophages carries out through the walls of the alveolus into the lymph nodes. And so these black deposits are actually lymph nodes on the outer surface of the lung. So the reason I'm showing you this is that when you have insoluble compounds which are coated with toxic materials, and these could be toxic organic materials, they could be toxic inorganic materials, but in a relatively insoluble form. These materials accumulate in the lymph nodes on the outside of the lung. They can stay there for decades. Now, what's really interesting is that the most insoluble material known to man, whatever it is, and the one that comes to my mind is plutonium, because I had some experience with that, but plutonium oxides are very insoluble. If they are trapped out, and they are carried out to the lymph nodes just like the carbon particles are, over years, and really decades, the plutonium migrates. Atom by atom, it'll dissolve, get into the bloodstream, and actually show accumulation in organs like the liver and the brain. The same thing happens with toxic organic compounds. And so there are a number of other routes that these toxic organic compounds that are originally on particles that might not have been able to get anywhere inside the respiratory tract if they weren't carried by these nanoparticles or other particles into areas where they have time to deposit and slowly dissolve, might not have as much of an effect. But the combination of particles and these toxic organic materials have a potential for affecting organs outside of the lung. So we can show that the material that gets inhaled does deposit in the lung, and occasionally, especially if you have very large doses, and this is something you see with miners, coal miners especially, there's a black lung disease, which is where the material deposits in the lung at such an amount that the macrophages are overwhelmed. They can't carry it out. And it becomes encysted in the lung tissue and forms fibrotic masses. And so black lung disease is really a fibrotic disease of the lung, which causes the lung to be stiff. Well, when we look at the lungs of people who live in urban environments, you can see beneath the lung tissue, in the interstitial spaces, accumulations of carbon materials. When this material gets into a cell, it can have a number of different effects in terms of toxicologic mechanisms. And so if it goes into the cytoplasm, material can dissolve in and cause reactions with membranes and various subcellular sections. One of the most important things that we've found, especially for the nanoparticles, is that they're small enough to not only get into a cell, but to actually penetrate through the walls of mitochondria. And so they can actually get in and damage mitochondria. Now, mitochondria is very important because that's the energy source for the cells. All your cells have them. And if those get disrupted, that cell will probably become non-functional. Now, for most cells, they have a relatively small number of mitochondria per total cell mass. Cardiac cells, on the other hand, have something like 70% of their volume are mitochondria. So if particles get to the heart, it can be very damaging. However, and this is one of the reasons that it's very difficult at this point to understand the real toxicology and the dose-response relationships. And hypothetically, if a few particles got into a mitochondria and was able to disrupt its function, the number of mitochondria, because you have a lot in a heart cell, if it were only a few nanoparticles, it might not have much of an effect. But because nanoparticles are present in huge numbers, so in this room, if we were to count particles, we would probably find tens of thousands of nanoparticles in the air, and per cc, tens of thousands per cc. Every time you breathe, you're taking in, say, 400 or 500 cc's of air. So you can do the math and you can figure how many nanoparticles can you actually bring into your body in a very short period of time. So if they're benign particles like sodium chloride or something like that, it might not have an effect. But if it's an insoluble carbonaceous material, it's not going to dissolve, that might have a potential effect. And this is just an example. This was some work done at UCLA. And what it's showing is this is, a low magnification or a high magnification of the same area. This is a cell, a macrophage. The macrophage has some mitochondria, and this is what a mitochondrion that's healthy looks like. If you feed the macrophage coarse particles, those coarse particles enter the macrophage and get collected in vacuoles. So these are little sacs of digestive fluids, really. And the macrophage is trying to eat these particles, but it really can't. But you can see that these particles, which were originally 2.5, and you look here, this is the scale. This distance is 100 nanometers. And you look down here, and you can see some of these particles have disassociated. So even the big particles, if they get in, have some nanoparticles associated with them. But if you use nanoparticles per se, just by themselves, again, they get into the cell, and you can see them distributing through the cell. But when you see what happens to the mitochondria, there are a few nanoparticles in a few of these mitochondria. But you can see that the mitochondria no longer have their functional structure anymore. So these mitochondria are effectively dead. And it's only from a few particles in each mitochondria. So the take-home message here is that particle mass may not be the best way to judge the effectiveness of nanoparticles. We might have to go to something else related to how many nanoparticles are going to be getting to a target, or maybe the surface area of the nanoparticles might be important. But it's something to consider. What do the nanoparticles that come out of combustion products look like? Well, this is a micrograph, an electron micrograph of a diesel particle, which is really a chain agglomerate. So a lot of little nanocarbon particles stuck together. But what's interesting is when you compare the structure here, or the appearance, to a manufactured carbon nanotube, you can see that there are some similarities. They're not remarkably similar, but you can see sort of a family resemblance, if you will. Now, the reason I throw this in is recently the NIOSH, the National Institute of Occupational Safety and Health, has been looking at health effects of carbon nanotubes. And they're considering reducing the occupational standard for carbon nanotubes to 1 microgram per cubic meter, which is a remarkably low level. And the only reason they set it at that number is that's the limit at which they can measure it. So they feel, at least in the occupational sense, these freshly formed nanoparticles have much greater toxicity than the bulk of elemental carbon. Now, diesel particles are not exactly the same thing and might not have the same degree of toxicity, but it's something that should be kept in mind. So do ambient ultrafine particles affect cardiovascular disease rates? So when you look at increases in air pollution on a national basis, and you associate particle concentrations with mortality and hospital admissions, there's a very clear association. Exposure to the elevated levels of particulate matter leads to physiological changes like increased heart rate and decreased heart rate variability. And this is especially important for elderly patients with coronary artery disease. Heart rate variability is an interesting function. We all exhibit heart rate variability. We have to. every time you move, it requires energy, which your heart has to supply the oxygen for. It's got to pump more blood. And so on a moment-to-moment basis, your heart is constantly adjusting for a perceived, it's not necessarily what is going on, but even a perception of a need. So if you get frightened, adrenaline kicks in, your heart rate has to go up. So you get variations. And I've seen this physically with patients with coronary artery disease when we were putting them through exercise testing and we had them on a bike. And you tell them, we're going to start the exercise test now, their heart rate immediately goes up. They haven't pedaled yet, but their heart rate goes up. So it's not just a physical need. There is a mind-body relationship here. So anyway, in order for your body to respond to day-to-day changes in stress levels, in work levels, and everything else, your heart's got to have the ability to vary. It turns out that one of the markers of impending disease is when your heart begins to lose that capacity. So we've been measuring heart rate variability in our studies as an indicator of possible adverse effects. And in our case, we're able to compare that to physical adverse effects like atherosclerosis. In terms of epidemiology, we find that, the elderly population above 65 years of age tend to have higher susceptibilities to air pollution-associated heart disease, especially and mortality rather. So all of this information tells us that we really need to look carefully at these particles. Now, in terms of numbers, in terms of public health, something like 50% of deaths on an annual basis are associated with heart disease. So we're really talking about a very large, a client population, if you will. From previous studies, we've been able to show that ultrafine particles are more effective than fine particles at promoting atherosclerosis, and we did this in some animal models, and I'll show you some of that data. We've also shown that some of the biomarkers, circulating biomarkers in the blood that are associated with cardiovascular disease in humans are increased in our animal models. So this tells us that our animal model is not perfect, but it follows the same pattern as humans. And again, we think that the reason that the ultrafine particles are really important is that they provide a very large surface area that can carry toxic components. So at UC Irvine, we've been very lucky in having a very integrated program. We've got an epidemiology group where Ralph Delfino has been looking at cardiovascular effects in humans and been able to associate biomarkers of change with changes in physiology. June Wu has been using land use regression techniques, and she's been able to show that traffic-related exposures seem to be very effective at promoting some adverse effects. And Rufus Edwards has been looking at various other exposure and risk assessments. So we've been sort of pooling our knowledge and using that to generate new hypotheses to test. So we have been testing the hypothesis that a near-road exposure would promote atherosclerosis. We had shown in the past that it helped to promote airway allergies. So there was a reasonable association and an assumption that we might have really good success in doing this project. And we were also lucky in being able to collaborate with an organized research unit at UCI called AirUCI, which has remarkably good atmospheric chemistry capabilities. And we were able to bring some of that talent in to look at our exposure atmospheres. And we've worked with our colleagues at USC and UCLA in doing more characterizations and developing our exposure systems. So the methods we use. We're going to try to test, and this project really tried to test, a specific mechanism by which PM exposure exacerbated heart disease. And we know that ultrafine particles do accelerate atherosclerosis in mice. And we've shown that if you take the ultrafine particles and remove the organic constituents, those particles are less able to have adverse effects in vitro testing. And so working with Kostas Siotis and his team at USC, we developed a system that integrated a particle concentrator, a thermal denuder system, and an exposure system so that we could generate high concentrations of ambient particles in a specific location. We could either expose the animals directly to the ambient particles or denude those particles in a thermal denuder and compare the results of those two types of exposures. And we could use a single particle aerosol mass spectrometer to do some very clear characterizations of what the particles look like. So for those of you who haven't seen a particle concentrator, this is the one that was designed originally by Dr. Siotis at USC, and it works by sucking ambient air in to a saturation tank. In the saturation tank, we have heated water, which creates vapor. The water vapor and particles are sucked up through these condenser tubes, which are kept at a very low temperature, around -5 degrees C. Under those conditions, the water vapor condenses on the particles, and the particles grow. And so the nanoparticles grow to a size where they actually have inertia. Now, to guarantee that what we're dealing with are nanoparticles, we have the air inlet go through an ultrafine particle impactor. So all the bigger particles are stripped off the airstream before they go into the saturation tank. So they come up out of this condenser, are focused into a beam, and shot into a takeoff tube. Now the takeoff tube operates at about 5 liters a minute, but the air is being sucked up in this direction at about 100 liters a minute. And we have 3 of them, so that's why it says 300. But what we're effectively doing is increasing the concentration of the particles by up to 20-fold. And as an aside, we're sucking off most of the gas phase materials, or a lot of it is removed by reactions in the tank, so that the air coming through here is pretty much enriched in particles. But things like ozone and nitrogen oxides are really not concentrated and are present at much lower than ambient concentrations. So the effects I'm going to show you are effects caused by the particles in the absence of things like ozone. So after the particles come up, they go through a diffusion dryer. And the particles are dried back down to ambient humidity. They return to their original, or very close to their original, particle size. There is a slight change in particle size distribution, so that what we get, if we looked at the particles in a particle counter, we might have seen in the ambient air a very broad distribution around the particle median of, say, 70 nanometers. when they come out of the concentrator, it's still a 70 nanometer particle approximately, but the peak is very narrow now. So they're really, the bigger particles and the smaller particles are all in the same size range. The thermodenuder is a device that we're using, and if you look at it, it's sort of similar to some of the afterburner technology being used in diesels. So what this does is the air enters the thermal denuder through a tube and is heated up. Then that tube passes into an absorption cooling zone. Now in this absorber, what we actually have is a large tube which is made out of activated carbon. So the particles go through the lumen of that tube, and they come through and aren't affected, but the vapors, the organic material that was on the particles, is volatilized by the heating and is in the vapor phase, and that's rapidly taken up by the surrounding charcoal. So when it comes out of the denuder, we have particles that are virtually stripped of most of the organic constituents. And then those can go either to chemical testing or into our exposure chambers. This is the Aerosol Mass spectrometer. This is just a cartoon of how it works. But it's a remarkable device, and what it does is it allows us to bring in particles, and this can be before or after they're concentrated or denuded. They go into this lens area where they're accelerated into a beam, and then they go through this chopper. And what the chopper does is it rotates, and so every few milliseconds a little pulse of particles come out. And the particles are accelerated by ionic forces, and the little particles, they all have the same initial energy put on them, so the little particles have a higher velocity than the bigger particles. And so they separate, and as they come out, the little particles come out further and further ahead of the big particles. They hit a heated plate, and then they're volatilized and ionized and shot up into a mass spectrometer. And the mass spectrometer uses, it's a quadrupole type mass spectrometer, uses magnetic fields and time of flight to separate the molecule fractions, and then runs them into a signal collector or a charge collector, and then a very complicated computer analyzes the data and tells us what there is. And that is part of the magic of this thing, which is beyond my pay grade, I think. But what's remarkable about this is it gives us a chemical analysis as a function of particle size. Many of you are probably familiar with the scanning mobility particle size spectrometer, the SNPS. It's used in many kinds of atmospheric studies, and what it does is it uses electrical mobility to collect particles as a function of size and analyze size distributions. So it doesn't do chemical analysis, but it counts particles and bins them by particle size. So this is how we knew that what went into the particle concentrator and what the size distribution of the ambient particles were, and we're able to compare that to what comes out of the particle concentrator. And that's why I can say that there's a very small difference, but it's not a major difference in particle size characteristics when they come out. The exposure system is something I'm very proud of because it took some time to develop this. In this system, we can put rats or mice and expose them to particles from the concentrator, from the denuder. And this system is built to be mobile. So we can actually rack up chambers with animals in them, roll them into a van, drive the van out to a site where we do exposure studies, and then bring them back to the lab so they can sleep overnight. While they're traveling, we use air purifiers on board so that they're only exposed to purified air. And when they come back to the lab, they're only exposed to purified air. But during the exposure, they breathe the polluted atmosphere. My technicians don't get to breathe purified air. So I'm trying to work out a way we can use them as part of the experiment, but I don't think it's going to fly. So our exposure protocol, these are mice that are called ApoE knockout mice. What that means is one of the genes that controls lipid metabolism, the ApoE gene, has been surgically altered. And these mice don't express the ApoE protein. So these ApoE mice, because they don't metabolize lipids very well, have cholesterol levels in the thousands. And almost all the lipids are low-density lipoproteins. So as you have probably seen from commercials on TV, these are the absolutely worst conditions for cardiovascular disease. So these are mice that are pre-programmed to be susceptible to developing plaque. So when I show you the data, you'll see that even the mice breathing controlled purified air atmospheres will have some plaque. We implant them with a miniature electrocardiograph. And this is something kind of like a pacemaker. And it's implanted inside the animal, and the body of the device is actually in the abdomen, and wires come out across the chest, kind of like the leads you'd put on for a regular cardiograph. The device also has a small radio transmitter built into it, so which we can turn on and off with a magnet. It's kind of strange, but that's, it allows us to save battery sometimes. But the mice actually are constantly broadcasting their cardiovascular signal. And we have receivers underneath the mice that report to our computer. And so we are collecting electrocardiographic data on these mice all the time that they are in the system, either when they're in the exposure system or when they're home in their cages. The only time we don't monitor is while we're driving them. So very early in the morning, we wake our mice up, put them in our chambers, load them into those rolling devices, and drive them out to, in this case, to USC, and we expose them for 6 hours a day, 4 days a week, for a total of 8 weeks. At the end of the exposure day, we bring them back home and put them back in their housing. So we monitor EKGs or ECGs for 21 hours a day, and of course, everything had to be approved by our University Animal Care and Use Committee. This is just to give you an idea of the location of where the area was that we were operating. So it's right near the 110 freeway and the 60 freeway. So an area, and we have winds typically from the southwest. So this area was a very good one for testing relatively close in freshly generated mobile source pollution. And of course the 110 freeway has no traffic on it, ever. It's practically a parking lot. So this is just a schematic to give you an idea of what we did. And what I'm going to report on were actually done as 2 separate experiments. The first experiment, we tested the difference between denuded particles and undenuded particles. The other experiment was sort of different. And there, what we wanted to look at was, because what we had seen here in the first experiment was a major effect of the organic materials. And we asked the question, well, if the organic materials on the particles have an effect, what happens if you just have the organic materials without particles? So we used the same denuder, but now instead of having a charcoal trap to collect the organics, we used a quartz filter trap to remove the particles. And so in this second experiment, we have a group that is exposed to something we called PFO, particle-free organics, and we compared those to the undenuded particles. And then we did a whole variety of various bioassays. We looked at lipids, we looked at the LDL cholesterol, we looked at C-reactive protein. These are all markers associated with cardiovascular disease. We looked at protein oxidation as a measure of oxidative stress. And we also looked at the amount of lipids deposited on the walls of the arteries. And then we did some microscopy to also show that we had plaque building up in these arteries. So what did we find? This is the kind of data we get out of the mass spectrometer. And what's interesting is the input to the mass spectrometer was the fine particles, so PM2.5. So you can see this went from 2 microns down to about 5 nanometers. And this dashed line represents about the 180 nanometer size of the ultrafine particles that we use in the experiment. And what we found was that, a few things. One is that the ultrafine fraction is very highly enriched in the organic materials, whereas the larger particles have sulfates and nitrates and ammonium, sulfate, ammonium, and nitrates, as well as organics. But the organics in the ultrafine fraction have less oxygen related to, with respect to the amount of carbon than the particles in the larger particle fraction. So here, there's roughly a factor of 10 difference in oxygenation, which has been reported in some respects that diesel exhaust, when you look at the oxygen to carbon ratio in diesel exhaust, it's more like this than like this. So this is a pretty good indicator that an important source of the ultrafine particles at this, at the site. And in this particular case, these measurements were not made up at USC where we would have expected to see lots of diesel. This was measured in Irvine, where we have some diesel traffic, we have a lot of automotive traffic. So we're seeing, even in Irvine, a very important fraction of organic carbon that seems to be related to these mobile source combustion type materials. Now what happens when you take the organics away from the particles? In the test tube, in vitro testing, we did a couple of things, but the most interesting was we analyzed the material for polycyclic aromatic hydrocarbons. And these were the high molecular weight ones. And we tried the denuder at different temperatures just to see what would happen. And it turned out that we could remove a little bit, a small amount, at 50 degrees C. When we heated it up to 100 degrees, we got rid of more than 50%. When we went to 200 degrees, we got rid of almost 80% or 90%. So the higher you heat it, the more of these organics you drive off, which stands to reason. Now, we actually did our experiments at 125 degrees, so a little bit over here. So we didn't expect to get rid of all the organics. Now, why did we pick that temperature? Because Kostas and his students did some very exhaustive tests on the denuder, and he found out that if you tried to run the denuder at different temperatures, at 125, we got all the material in the vapor phase that we could deliver to the animals, and no particles nucleated. So that's why I can say that that was particle-free organics that we're exposing them to. So doing the experiment really required a lot of front-end work. What did it do to the particles? Art Cho and John Freunds at UCLA used their DTT assay, which is a dithiothreatol assay, which tests for the ability of the particles to generate free radicals. And you can see that as you remove the organics, you remove a large percentage of the ability of the particle to generate free radicals. So intrinsically, this is saying that these might be less toxic. These are the actual data from the experiment, and it's important to look at the difference between the denuded and the undenuded particles. So the first thing I want to point out is that when we denuded the particles, we lost about half the mass. Now, that could be by a lot of reasons. You could have all sorts of things going on. But when you look at the organics, the organic carbon, you drop the organic carbon to about 1/3 of what was originally present. So a large fraction of that loss of mass is because we've stripped off the organic carbon, which is exactly what we were trying to do. So there is some loss of particles, but we do have an explanation for most of it. When we look at the inorganic constituents, so chloride, nitrate, and sulfate, the only one that is significantly changed, and there are differences between the measurements, but when you take into account the detection limits and the errors involved making the measurements, only the loss of nitrate, which was not unexpected, was statistically significant. When we look at the polycyclic aromatic hydrocarbons, we find that the ones that are removed are the pyrenes and the benzo additive products of pyrenes, which again, these are the ones that people associate with the formation of free radicals, with the formation of a number of other things and health effects like cancer. And they've even been associated with proliferation of smooth muscle cells. So these are, biologically important materials. And these are the ones that were very greatly removed, including the pyrene. The rest of the polycyclics where we had adequate measurements, you can see, phenanthrene was not really reduced. But in other words, the differences between the denuded and undenuded were not statistically significant. So it's these benzpyrenes and a few others that we are very effectively removing. What does that do to the effects on the particles? So these are heart rate variability data, and we have different measurements of it. And it's clearer over there than it is on the computer. Okay, so this one here represents the high frequency component of heart rate variability. When you look at heart rate, you have some fast beats, some slow beats. We use spectral analysis to separate out the high and low frequencies. But what you can see is the green line is the undenuded particles. And you can see clearly after the second week, the undenuded particles have lower heart rate variability than either the air or the denuded particles. So denuded particles are not significantly different. The other thing that is subtle and really shows up best in this total heart rate variability, where we look at sort of the combination of the high and low frequency components, you can see that over the 8-week period, there's really a progressive loss of heart rate variability. And we looked at a separate measure, and this is the relationship, the low frequency to high frequency ratio, to try to get an idea of what was going on. And it turns out that the low frequency increased, the high frequency decreased a little bit. The net result is the low frequency and high frequency of the undenuded particles. Did I have that right? Yes. And the denuded particles are different from the air. So both of these components, even though the undenuded particles didn't really cause a progressive change or a difference from the denuded particles in terms of overall heart rate variability, they still have an effect on the autonomic nervous system. So I don't know exactly what that means yet, and we're going to work on that some more. But I think it shows that even the denuded particles may be playing a biological role. Now this is a little bit difficult to look at, but I want you to take a look at this as sort of a heat map. And what we're measuring, these are the data from air animals, undenuded particles, and denuded particles. And just from looking at it, this is what happened, what was measured in these animals before we did exposures. So this is the week before, when we measured over a 10-day period, and you can see that there are sporadic variations in heart rate variability. When we started the exposures, you can see that the animals that got the concentrated particles in the undenuded form, heart rate variability, which we're measuring here as animals showing more than a 50% decrease. So it's a substantial amount. You can see that as we began the experiment, there wasn't very much going on. But as the experiment went on over the 8 weeks of exposure, and these are just broken out in days, so it says 30 days, you can see that over time, the effects got greater. And the effects, now this is, you can sort of make this out, and this area here where there's very little change, this is the period when we're actually doing, this part here with the change is where we're doing the exposures. Over here is where we're moving the animals. We don't have very much going on. And then when they're in their cages at night, you can see that there's something that goes on right around here. That's when we turn the lights on. These are nocturnal animals. And they're in the dark during the day. We turn the lights on at night because that's when they wake up. And so there's a bit of a peak. And you can see a little bit going on in the animals with denuded, but the undenuded guys have a continuing effect. And it gets progressively worse over time. So I think that's really an interesting picture. This is something we just generated last night. So it's actually not in the broadcast material, unfortunately. All right, what happened with other markers? So here we're comparing the denuded to the undenuded particles, and we looked at serum LDL. Interestingly, the serum LDL was increased, and only the ones with the denuded actually got to statistical significance. There was a little bit extra noise in here for some reason. But when we looked at lipid peroxidation, because it's the peroxidized lipids that end up in the plaque, only the undenuded material significantly increased lipid peroxidation. So that even though the LDL levels were comparable, the oxidized levels of lipids were much greater in the undenuded exposed animals. Now I want to point out that this is not oxidized LDL. We weren't able to measure that directly. We didn't have big enough samples. So this is total lipid peroxidation. We also looked at the percent of plaque in a couple of different arteries. And I'm going to show you here samples from the aortic arch. And the aortic arch is the part of the aorta that comes out of the heart and flips over and then goes down to the lower extremities. So this one here, the top one is an air-exposed animal. You can see a very typically smooth, narrow wall and an empty lumen. The animal exposed to the denuded particles, the walls are a little bit thicker than here, but again, the interior of the lumen is relatively clean. When you look at the undenuded particles, the walls are thicker, and you can really see that the interior lumen of the artery has been greatly decreased. So the denuded plaque may have an effect on arterial walls, maybe causing some inflammatory process, calling in other cells, or maybe even stimulating proliferation of the smooth muscle cells that make up part of the wall. But the undenuded particles that have the organics on them clearly increase the amount of plaque deposited. Now, when we looked at the particle-free organics, we found that they also changed the low-frequency to high-frequency ratio to a greater extent than the undenuded or the purified air. And when I just plotted this up, let me see it, grow it. Yeah. So when we look at heart rate variability, this is the particle-free organics. And you can see that this does tend to decrease heart rate variability. And also the undenuded particles tend to decrease heart rate variability. But in this experiment, the heart rate variability in the control animal sort of stays about the same level, a little bouncing around. But we're seeing an effect of the particle-free organics. So when we looked at things like serum LDL, again, we see very similar effects of the particle-free organics and the total. We see on the total LDL, again, total and the non-volatile fraction, they're about the same, a little bit more than the air. Take off my glasses. The total cholesterol in both animals, both groups of animals, are relatively high. But when we look at the cholesterol and the PFO in the second experiment, there is a significant increase. When we look at peroxidation, both the undenuded particles and the particle-free organics cause lipid peroxidation, whereas the denuded particles, this is the same graph I showed before, do not. So conclusions. I think one of the important things is we've now shown that we can actually use this system to do some very interesting biological studies. And we can not only generate denuded and undenuded particles, but we have the capability of using particle-free organics as well. We find that the undenuded particles accelerate the development of the atherosclerotic plaque, and that the exposures to undenuded particles and to particle-free organics alters heart rate variability. And we have to conclude from the study that the organic constituents are playing a very important role. We found that both the organic and inorganic constituents, for some reason, both raise serum lipid concentrations, but only the particle-free organics or the ultrafine particles undenuded oxidize lipids. And it's the oxidized lipids that are the ones that are active in forming the plaque. So by analogy, I think that thermal denuding techniques, such as afterburner emission controls, can not only reduce total pollution, but may actually change the toxicity of the emitted particles. And so this might be a rationale for looking at improving those techniques and exploring how to use them more in the environment. So I want to thank, the ARB for a major part of the support, but we also got help from our friends at AirUCI and NSF. We got some support from US EPA and NIEHS, and we've had a whole team of players. I've only picked out the 5 individuals that actually helped produce the numbers that I showed here. There are many more people involved. But Dr. Siotis at USC, Andrew Keebaugh is one of my graduate students, Lisa Wingen, who works with Barbara Finlayson-Pitts on the atmospheric chemistry, Payam Pakben, who has been doing the denuder work at USC and generated the initial data on that and taught us how to use it, and Loida Mendez, who did a lot of the biological assays. So I'd like to thank you for listening, and if you've got questions, I'd love to try to answer them. Yes?
Yeah, I had a couple of questions. One is sort of scientific, the other is a little bit personal. But the scientific one, like what percentage, and does it vary by the type of particle, how big they are, like what percentage would actually be retained in the lung from the different?
It varies with size.
Yeah.
And so, for example, particles around a half micron, thereabouts, don't deposit by diffusion very well, don't deposit by impaction very well. So a lot of those that you breathe in, you breathe out. So a very low percentage, maybe 5 or 10%. Ultrafine particles, almost 100% will deposit somewhere.
Wow.
The fine particles, the PM2.5, about 30 to 50% will deposit in somewhere in your chest. And the coarse particles, something like 30 to 50% of those will deposit more in the higher bronchi and in your head. So varies with particle size.
Okay. My other question partly relates to the building which I work, which is east of here. What percentage of the particles that say would be out on the freeway would make, if your building was right next to the freeway, what percentage would would end up inside the building? Or building, say an office building, very effective at siphoning out those particles?
Again, depends on particle size. So just like your lung, these particles, like the half-micron particles, thereabouts, aren't stopped by much. They'll drift right on through unless you've